Okay, great. First of all, thanks everyone for joining us and for allowing us to do this combined presentation. This work is really an equal collaboration between my lab and Antonio's lab, and it's spearheaded in each of our labs by two really talented postdocs, Henry Shun Chan in Antonio's lab and Candice Wyke in my lab, who've really taken this work forward in each lab. I'll say in my laboratory, Candice is the only person doing architecture, and in fact, she's the only person here at Utah who's doing it, so she's had to pilot all of the methods and the computational biology, and she's really appreciative of the 4DN network and all of their help. That's really enabled us, I think, to do the work on our part that I'll show today. So the question that we really have is to understand the relationship between transcription and nuclear architecture in the very early embryo. And the early embryo is a unique time in an organism's life because after fertilization, there is a period of transcriptional quiescence, but the genome and the chromatin of that genome are being arranged to prepare the genome for transcriptional activation. And the length of that transcriptionally quiescent phase, which is shown in this slide you're looking at now, is different in different organisms. So the red period is the period after fertilization where the transcription is quiescent. The light blue section next to it is called the minor wave, where there is a subset of genes that are getting activated. And in most organisms, it's a particular subset, and it is as well in the fish, as I'll be showing you. And then on the right side, in the darker blue, is the major wave of transcription, where the vast majority of the genome wakes up. So the question that we're trying to address is, what is the relationship between architecture of the genome and transcription? And we'd like to understand by genomic methods, what are the first transcripts that come up in those minor and major waves, and what is their relationship to architecture? And can we have a combined genomic approach and visualization approach? Can we both monitor them by genomics and also have sophisticated imaging techniques to be able to look at the loci that are transcribed, both by marking the DNA visually and by marking the nascent transcripts at that time? So in the top section here, we're focused on the zebrafish because the talk today, and Antonio in my lab, is focused on this question in the zebrafish. So at fertilization, there's no transcription, and then the green indicates the phasing in of the green indicates the phasing in of transcription, which the minor wave is starting around the 256 cell stage, and then the major wave is starting between the 1,000 cell stage and 50% epithely, but really starts at that 1,000 cell stage. So we'd like to, what the zebrafish offers is an opportunity to uniquely look at this minor and major wave. This is something that's very difficult to look at in the mouse, for example, because you only have one cell cycle before you start transcription. So this is very difficult to look at in the mouse by genomics methods, etc. But the fish offers this wonderful period of about 10 cell cycles, where you have 5 or 6 cell cycles before you get to the minor wave, and another 5 cell cycles before you get to the major wave. So you can, both by genomics and visualization techniques, look at this problem in detail. So that was the collaboration that we formed. My lab focusing more on the genomics, although Antonio's lab is very good at aspects of that as well, and his lab focusing on the imaging techniques. So in the following slides, Antonio is going to go through the imaging properties and some of the advantages of the zebrafish in looking at the first transcript that are transcribed in the zebrafish. Antonio, take it away.
Hello, everybody. So thank you, Brad. As you can see, this is a true collaboration, even when we present our work. So this is actually representing a zebrafish embryo, where you can see the nuclei label, and this is just to give you a sense of the early developmental stage when we are looking at the activation of transcription, where it's really characterized by the rapid cell divisions after fertilization. So what we have begun doing is combining, on our side, imaging approaches at the single cell level with genomic approaches from Brad Laboratories. So what you can see here is that within 4 hours of development, you can see the rapid subdivision that has happened after fertilization. So if we just stop this movie, you see basically what we do is that we inject a clicked nucleotide to then label the nascent transcription. And what I'll be showing is just the image of a single nucleus, and all this work has been done by Henry Chan, and it's unpublished. And what we did was basically to understand when transcription really begins in the early embryo, we backtrack in the early stages, and you can see here 3 different stages where we are labeling transcription at a single nucleus of that embryo. And what we can see is how this initial wave of transcription happens in just 2 loci, labeled here in yellow, suggesting that this particular locus is the very first locus that gets activated in the genome, and then you have gradual activation of the genome, and then you have the global activation of the genome about an hour and a half later. So the very first question that we asked, well, what could be this very first locus that is activated? And we had some clues from our previous work where through gene expression analysis, what I'm showing here is a genome track, we observed that the MIR430 locus is highly activated very early in development. So just be aware that these are different scales. So you can see initially there is very little transcript level for this microRNA. And then later on you see gradual activation of this microRNA that is composed within 17 kbs of 50 copies of this microRNA. And you can see how the scale dramatically changes between 256, about 2.5 hours, and 3 hours, when there is a massive explosion of transcription here by the change in the scale. Indeed, we have now seen that this is labeled strongly labeled by H3K27 acetylation and H3K4 trimetylation in this locus. So we asked, could this be the locus that we are actually visualizing here through nucleotide incorporation into the DNA? Also RNA into the RNA that is being transcribed. So we use the system from Bo Huan and Jonathan Weissman with dead Cas9 labeled with 3GFP, and we use 2 guide RNAs that basically are able to recognize 20 sites each within the MIR430 locus. And lo and behold, we found that, in fact, this locus that is the very first actively transcribed locus within the zebrafish genome coincides with the MIR430 locus labeled here by the GFP. And as a control, we use a deletion mutant that lacks this locus, AKB deletion of the MIR430 locus, and you can see that we have no signal. So at the moment, or following the identification of the very first actively transcribed gene, one of the goals is to basically understand how the interaction and the formation of the different chromosome regions and the formation of different TATs influences transcription. And to address that problem, the very first goal was to really understand whether we can really visualize transcription in vivo. So to achieve that, we use a molecular beacon that is actually containing a quencher in one side and a fluorophore on the other side, and this forms a hairpin that quenches the fluorescence. And we injected them in the embryo and asked whether we can visualize transcription in vivo. So in this case, what we are seeing is a 64-cell stage. We're focusing on a single nucleus where you can see the molecular beacon in red in this insect. And what we can see is that in a living embryo, we can see the burst of transcription here over time. And this was quite exciting because it's the first time that we can really visualize transcription in vivo in the zebrafish embryo, directly looking at the hybridization between this oligo and the transcript, the primary transcript that is produced from this locus. So in the future, what we are planning is to use higher resolution microscopy to label transcription as well as the different TATs that Brad is identifying through Hi-C to then determine how the interaction between different regions influences the transcription within this locus, the very first locus of genome activation. Oh, sorry. So Brad, you want to take it away?
Okay, sure. So in the next section, I'll be telling you about our efforts to understand the 3D chromatin architecture using high-C methods to understand how does the architecture develop, on what time frame, and what is its relationship to transcription. Is it going to be similar to what others have seen in the fly, for example, or will there be unique attributes or differences in the vertebrate fish in how this is accomplished in those relationships? So to that end, we have been doing isolating embryos for high-C. Go to the next slide, please. And I'll just talk about the time points that we're using for this effort. So we're going to be looking initially at ZGA, which is at zygotic genome activation, which is at 1,000 cells. And then we're going to be looking at 50% epibole, which is 5.3 hours. And then we're going to be looking at 24 hours, which is a control of later in development. Of course, ideally, we would like to look pre-ZGA, which will be 256 cell embryos as well. But since we have not done high-C in the fish before and it hasn't been worked out, we're piloting it on the ZGA and later levels. And once we get that working well, and as I'll show you, I think we have some preliminary results that look promising, we're going to go back and do these much more difficult earlier time points. So we want to look at the relationship between transcription and architecture in a wild-type fish to begin with. And then we will be using drugs to inhibit transcription to then see if what the relationship is between architecture and transcriptional activation. And then we'll be doing some functional studies by doing protein elimination in the early embryo to look at the requirement for certain protein factors in establishing that architecture. So as I mentioned before, these are our 4 time points. I only have the latter 3, 4 hours, of 5.3 and 24 hours now, but we do have preliminary data. If you look at the table to the right below, we have about 40 to 50 million mapped reads total for these. This is obviously not deep enough to do detailed analysis, but it's deep enough to do pilots to let us know how well we're doing. Our intention now is to go deeper and sequence about 3 or 4 times this level to get our final data set. But I'll just preview to you what we have now with this preliminary data set. I'll just say that if you look on the left, the contact probabilities look reasonable in terms of the initial data. If we go to the next slide, this is the initial high-C maps visualized by high glass. So we're looking here at a portion of chromosome 11, for example, just to illustrate this. And what you can see at this modest resolution of sequencing is that we do have some architectural features that can be observed even at 4 hours post-fertilization, so at ZGA. And then if you go forward in time, 5.3 hours and then 24 hours, those become more and more refined in the embryo. And but we don't have the resolution yet to clearly call TADs, but that is the ultimate purpose. But we are piloting that by using 2 different TAD callers. The high-C, high glass here, and then on the next slide, we have the high-C Explorer. And as you can see, it is calling architectural features here. And they are, at 4 hours, we can see some of those features that are being called. At 5.3, there are additional features that are emerging and changes. And at 24 hours, we can see more refined and higher resolution features. But again, I think we have to consider this as preliminary data that awaits more sequencing. We also think it would be, we can also tell that it would be very helpful to have nested TAD callers on this, because we can definitely see some architectural features inside other larger ones that would benefit from a nested TAD caller. So on the next slide. We're zooming in now on the locus that Antonio was talking about, this repetitive MIR430 locus that has the first transcription in that minor wave. And this box that Antonio is now trying to circle for you, this white box, includes the MIR430, which is right at the base, which if you move to the right like he has, that outset, you can see the MIR430 locus is right in the center of that white box and appears to be at the boundary of two architectural elements. And then if we look even at 4 hours post-fertilization, which is the time when the MIR430 locus is active, if we look at 5.3, there's a change in that region. And remarkably, once transcription ceases in that region, we no longer see a boundary, what appears to be a boundary element at that location. So this is preliminary, but we are hopeful that we are seeing a change in architecture at 430 between its active state at 4 hours from 2.5 to 4 hours post-fertilization, and then it's a change as its transcription gets shut off later on. So we also have preliminary experiments to look at the relationship to transcription, and those are coming up next. And what we've added here are inhibitor of transcription at 4 hours post-fertilization. So we eliminate Pol2 transcription. And if you look at the MIR430 locus at the right, we still see it present as a boundary element. So this might be consistent with the boundary not relying on transcription per se, but it might rely on the chromatin features, modifications, bound factors, et cetera, that might help establish that boundary at that time. But again, this is preliminary data. If we go to the next slide, in order to help us normalize and understand our data and to know when we have a negative result that's biological versus a negative result that is technical, we are engaged now in spiking in Drosophila chromatin into our zebrafish experiments. So we're taking these cells and spiking them in at a low ratio, a ratio that's high enough that we can detect them in our maps, but not so high that it swamps out all our zebrafish maps. So you can see on the left, for example, at the end of the day, we would like a map of the zebrafish, and then at the right, by mapping to the Drosophila genome, a normalization map of Drosophila, and that's an all-by-all comparison that's shown here, to which we can normalize these, our zebrafish data. So we don't have experience with this yet. We have done some preliminary experiments of our own to see how this is working. So for example, these are 4 experiments that both look at zebrafish embryos at 4 hours post-fertilization, so zygotic genome activation. At the top left is untreated, and this is the then we're looking at the Drosophila genome here that was spiked in. It's an all-by-all comparison. On the right is a DMSO mock, because the drug's in DMSO. In the bottom left is flavopuridol, which is the Pol2 inhibitor that we're using in DMSO. And at the right is the all-by-all examination of just the Drosophila S2 cells. So we think we are on path to be able to use the Drosophila data for normalization, but the next slide is kind of a call out on the collaboration plaza. If other people have experience with using spike-ins and would like to collaborate with us, we would welcome a collaboration with somebody else in the network to help us understand how to best apply the matrices, et cetera, to get this right. So Candace has put this up on the plaza, and we welcome a response to that and your comments if you have them. And on the next slide, so what we'll move to now is a discussion of what we're trying to do at the functional level. So the next phase once we get the maps of wild type and maps of dependence on transcription, is to say what is the contribution of particular proteins involved in loop formation, such as cohesin, or certain boundary proteins, such as CTCF and others, to this process in the minor wave and in the major wave. To this end, we're taking two types of approaches to address this. So I'll just say that we cannot take genetic approaches to eliminate critical proteins like RAD21, cohesin, or CTCF, because we will not get fish if we do genetic nulls. And we have to do a protein destruction type system to accomplish these functional experiments. So Antonio's group has developed a Auxin degradation system, which can effectively be used to destroy proteins. So here is an example of a auxin inducible degradation tag added to GFP, and in the absence of ligand, you see green fish, and in the presence of ligand, that protein is eliminated in the fusion protein. So this system is still in development. It's working, but needs some additional development to be working well and in all cases. But I think we're on path to using this system for our functional experiments. And in the next slide, we of course would like to use place-seq and chip methods to complement the other experiments here. And there are not, however, a good collection of zebrafish-specific antibodies to many of the proteins that we would like to examine. So the strategy that we're taking is to have a combination tag, a 3x auxin-inducible degradation tag on a protein coupled to a triple HA tag. And that construct can be integrated into the genome using a color marker. And then that color marker is a Venus protein linked to alpha crystalline, so that will be expressed in the eye of the fish. And then the correct recombinants will have a green eye. And we are collaborating here both with Antonio and also with David Grunwald and Kazuyuki Hoshijima here at Utah, who are one of the experts in doing recombination in early zebrafish embryos. And we are having some preliminary success on these methods of integration and tagging. And our plans are to move these even further forward with those two groups so that we can have in vivo tagged versions of these to do both chromatin IP and to do place-seq approaches. So in summary, I think we've told you giving you an update on our efforts to understand the transcription architectural relationships in vertebrate embryos. I think fish are provide a unique opportunity to look at how you sculpt the chromatin genome prior to transcription for transcription, and there may be differences between the first wave, that minor wave, and the major wave. And especially interesting is the ability to look at this particular locus, this microRNA 430 locus, which is important biologically for its use in creating microRNAs that destroy the maternal RNAs. And this combination of being able to both monitor it by genomics and visualize it, we think is quite promising. So we have preliminary high-C work that we have to, I think, get more sequencing for to really refine our understanding of the architectural aspects. We've piloted our transcription inhibition experiments, and we're very interested in this possible result that we have at MIR430 on its independence from transcription for forming a boundary element. We have spike-in controls because we would like to have confidence in both our positive and negative results, and internal controls are a great way to accomplish that. And so we welcome advice on that. and then to do the functional experiments and the genomic experiments, we're I think, fairly far along in doing both tagging and in auxin-inducible degrons to eliminate proteins. And with that, I'll thank you for your time, and Antonio and I, we'll take questions. Once I thank the people that did the work. This is all of our laboratories here, my lab and Antonio's lab. And again, perhaps Antonio can point at Henry here to say thanks to him, who has done all the work in Antonio's lab that we presented today, and to Candace, who did all the work in my lab that was presented today. And thank you for your attention.
Thank you.